Hello, everyone. So today we're going to go over with a unit after cardio, which is going to be respiratory. So first, respiration is known as two different processes. One is known as external, and the other is known as internal. External refers to the exchange of oxygen and CO2 between blood, lung tissue, and the external environment, whereas internal refers to exchange of these things between blood and body tissues. The primary function of the respiratory system would be based off of external respiration, basically physically moving air in and out of the lungs. The cardiovascular system and the skeletal muscular system are also critical roles to the physiology of external respiration. For example, the pulmonary circuit of the cardiovascular system brings blood to lungs for oxygenation, and the thoracic and abdominal muscles provide movement for ventilation. So, could say for ventilation, we have thoracic and abdominal muscles have cardiovascular, blood to lungs for oxygenation. It also has secondary functions in being a protective barrier, sensory, and communication. Now, for the upper respiratory tract, we have functions in terms of air conditioning, olfaction, which is smelling, and sound articulation. You can think of the upper respiratory tract as anything from the nose to down to the pharynx. The lower is going to be what's going to be composed of the larynx and down to the bronchioles. As for the lower respiratory tract, we have air conduction, moving air, and phonation, which is making sound, and external respiration. Now, the lower respiratory tract can also be divided into sections that conduct air and permit respiration. Now, conducting is basically moving air, and this is from the larynx to the trachea to the bronchi. There are two types. There's three types actually. There's first going to be first divided into primary bronchi, the lumbar bronchi, and segmental. I guess you can use PLS as an acronym, and then two bronchioles, which are just into terminal bronchioles. And then this would be the conducting portions. And as for the respiratory portions of the pulmonary lobules, which are essentially just the bronchs and alveolar sacs associated with them, and then an alveolus, which is a capillary wrapped gas exchange structure. This is going to be what's the conducting portion is going to try to move air downwards, and the respiratory system is when you have actual gas exchange. Most of the respiratory tract is covered with respiratory mucosa, which is something part of the immune system. You can imagine a sort of gel that has protective specializations. And yeah, they can trap condition air, they can trap pathogens, they can also condition air. And with cilia, they can move to the pharynx. So most of the parts of the respiratory tract covered by these mucosa have cilia that can beat it towards the pharynx, and then you can swallow them later, called the mucociliary escalator. But by the terminal bronchioles, we don't have any more. We don't have any more of these mucus or ciliated cells. So no mucus or cilia at terminal bronchioles. Instead, you have mobile macrophages that are present. And respiratory surfaces are covered with an epithelium that is specialized for rapid gas exchange. Now, in the alveoli, there's a thin layer of type 1 alveolar cells that line the surface. There's also type 2 alveolar cells, and they make surfactant which reduces surface tension. That's kind of the gist of how it is. External respiration only happens in the alveoli. Something we should really note. External respiration, alveoli. And the rest of the respiratory tract is simply just a pathway for air to flow in. Now, this is kind of different with our definitions of respiratory and conducting. In the respiratory portions, you'll see the alveolar sacs and respiratory bronchioles, whereas the conducting portions are going to basically have just be other portions, I suppose. Mm. And the challenge of respiration is that you need fresh air. However, the respiratory tract is a passive structure. So essentially what the body does is you make a pump. First, you have a double layer membrane called pleura that adheres each lung to the wall of the thoracic cage. And you'll have a periodal pleura, and you can also have visceral pleura. Changes in the size of pleural cavities create pressure gradients. So inhalation, if you want to inhale, you're going to want to actually have a negative pressure inside, so that pressure outside can enter in. So, and for exhalation, you want the pressure to be higher inside, so that air can enter outside, so the pressure outside is lower. And in doing so, you're moving the diaphragm. And basically for contraction, you'll you would need contraction to create a negative pressure gradient and relaxation for exhalation. Very small movements in intramuscular pressure can create movement of air through the respiratory tract. And another thing is during quiet breathing, only primary inspiratory muscles active. These include intercostal muscles and the diaphragm. And during active breathing, accessory muscles are needed. This involves the transverse abdominis and abdominal muscles. And the internal intercostals. Respiratory system movements depend on skeletal muscles, and thus we need somatic motor neurons. And the phrenic motor neurons helps innervate the diaphragm, keeping you alive. This is C3 to C5. And the respiratory cycle depends on neural activity generated within the medulla. If you have no medulla, you're not going to have any breathing. And they make synapses onto spinal motor neurons that drive breathing activity. The pacemaker is called the Pre-Botzinger complex, which drives patterns in the inspiratory and expiratory centers. Breathing in and out can be measured using a spirometer that measures the volume of air moving in and out and how fast it's moving. And some terms you might know that you might see from the spirometer. If we first have a spirometer here, you can see that this is how you're breathing normally. This is, let's say the doctor asks you to breathe in, then breathe out, or I guess breathe out and breathe in. Then these are the values that was like so. So let's draw some lines here. So this value here is going to be known as a tidal volume. This is going to be a tidal volume within how you breathe normally. This is going to be the inspiratory reserve volume. And the inspiratory reserve volume is going to basically be like, oh, hey, how much can you breathe in at most? And this measurement here that goes from the VT to IC is going to be known as inspiratory capacity. This is how you're going to be, what you're going to be looking at if you're, let's say, breathing in. And IC you can see is equal to IR inspiratory reserve volume plus the tidal volume. And then we also have the concept of expiratory. Just as we have the inspiratory reserve volume, we have the expiratory reserve volume. And then we have a measurement known as the vital capacity. The vital capacity is going to be equal to the IRV plus tidal volume plus ERV. This is just basically everything that you can really measure. And this is measured after um, maximum inhalation. And the residual volume is going to be what's remaining. And this one cannot be measured directly with spirometry. And with this, you would also have a measurement called the functional residual capacity, which is everything below from the tidal volume to everything that's in. So functional reserve capacity, which is the air left in lung at the end of the regular breathing cycle. And essentially, you can also have a measure called total lung capacity, which is this entire thing. Total lung capacity is equal to IRV plus VT plus ERV plus residual volume. Or you can say it's equal to VC plus residual volume. They're just some basic analogous ideas of the respiratory system, of the respiratory duct and all this. Some ideas that you should essentially know for any sort of exams. Now we can talk about ventilation. Respiratory reflexes alter our breathing patterns, and different sort of stimuli trigger different changes to breathing patterns. Metabolic reflexes are activated from chemoreceptors in elastic arteries in the medulla. If we remember correctly from last time, the chemoreceptors are known as corroded, uh, corroded arteries and, corroded and aortic bodies, whereas the basoreceptors are known as, and not basal, but bara, baroreceptors are known as arter, aortic arteries, I believe, aortic arc and corroded sinus. I believe so. Let me double check. Corroded sinus, aortic. Yeah, it's a corroded sinus and aortic arc. That's for baroreceptors. Anyway, see. Let me see. So, as for these metabolic refractory reflexes are made by chemoreceptors. Let's write that down actually. Chemoreceptors, corroded and aortic bodies. And they're more sensitive to changes in PCO2 than changes in PO2. And this stimulates respiratory rate and volume. So a sigh, one thing is a reflexive pattern, helps reinflate pulmonary lobules. And yeah, this is, if you don't have these sighs, you will likely have collapsed lung with this sort of case. And protective reflexes are also powerful reflexes to remove irritants. You have sneezing, that's part of the nasal cavity. That's stimulated the nasal cavity, and you also have coughing, which is triggered by things in the lower respiratory tract. The key thing is sneezing is involuntary, whereas coughing is not involuntary. Well, it's voluntary. Well, you can kind of control it. And coughing also remains closing of nasopharynx, which is something is key. The tidal volume is a small fraction of the total lung capacity, as we mentioned. Also, respiratory activity it needs to be matched to current oxygen consumptions. So one thing is our respiratory minute volume. We can have a measurement for the volume air moved in minutes. This is equal to the respiratory rate times the tidal volume. And if you want to compare it, this is very similar to uh, the cardio equation for cardiac output, except you change the measurements a little bit. The conducting passages have an anatomic dead space, which leads to mixing of used and fresh air. So if we look at the lungs like this, if we have air stuck in here, so this is because a portion of air remains in conducting regions. And since it cannot contribute to ventilation, we call it anatomical dead space. And usually the anatomical dead space is the vital volume times 0.3. And it's estimated from the regular tidal volume. And this makes it such that our equation for the alveolar ventilation is going to be equal to F times FT, oh sorry, the tidal volume minus the dead space. And you're going to have to likely have some calculations based off of this. You need a lot of energy to basically inflate a lung, but there's two components that we need to mainly consider. One is resistance, which is known as force to move air in and out. And the other thing is compliance, force to expand walls of the lung. So compliance is how much pressure to expand the lung at a given pressure. And one idea is how much, like if you have a balloon like this, how much energy or force it take to expand this balloon or make it bigger. That will be known as how much compliance you get at a given pressure. And it can refer to pressures of the lung tissue on its own. So we can mark lung tissue, but it can also refer to skeletal muscular elements. So let's say because your lungs are surrounded by the rib cage, let's say the rib cage is very inflexible and it's not letting the lung move out. That would increase compliance. And respiratory restrictive lung diseases are diseases of reduced compliance. So I should say restrictive. And these are the changes in the respiratory system or the thoracic cage. One idea is respiratory distress syndrome or arthritis. So yeah, uh, reduced compliance, meaning they need more force to expand or inflate the lung at a given pressure. Should I say at a given pressure? Now, restrictive lung disease is detected through reduced functional FLC or total lung capacity. And how you can kind of see this is force vital capacity. Well, remember our measurements for vital capacity was essentially like we're to measure like this. This is our vital capacity. Well, if we if we have difficulties both inspiring or expiring, you'll probably see lower rates like this. Like before, it looked like this, but now it's like here. So we should see a lower functional FEC or TLC. Oh God, I'm really bad with memorizing. Uh, Functional vital capacity or forced vital capacity reduced. I think I should call it forced vital capacity or total lung capacity. I guess you can also call it that. And yeah, let's see. Lower capacity for air crossing. And resistance is force needed to move air through conducting pathways. And this is similar to resistance of blood flow. And a biggest determinant is diameter of airway passages. And this leads to obstructive lung diseases and to increased resistance. And this could be asthma or also chronic bronchitis, of which the conducting pathways are obstructed, where you need more work uh, to get air to and from the lungs at a set time. Now, one example of, well, they are detected in spirometer by doing changes to the force expiratory rate. Remember how we have this? So if the force inspiratory rate expiratory, or I should say inspiratory reserve volume, expiratory reserve volume, if we see a change of which we have first, second is the way that what you're going to see instead of this change is you're going to see something where it's going to look a bit like this. It's going to take a longer time to get air in to catch up and fill the lungs. So the first second of forced expiration identifies changes in resistance. So imagine you're trying to breathe out, and obviously obstructive would mean it takes more work to get air in and out of the lung at a given time. So if it's taking you more time or more energy to get out, I should say not the same volume of air, but if it takes you longer to get out the same amount of air, you might be coughing and all this, then this could be an indication of this obstructive lung disease rather than restrictive. And emphyema is destruction of lung tissue, of respiratory tissue, especially elastic fibers in alveoli walls. And this is caused by smoking, and this leads to increased compliance, but decreased recoil. So this means that they are actually easier to inflate. But because we don't have a recoil, exhalation is harder. Because after you because exhalation is a passive process, but because we lost the ability of elasticity, they won't exactly like move back as calmly. Or I guess if that makes sense. Aphamesia can have similar effects to increase resistance. Because remember, resistance was about moving a given volume of air, the amount of energy it takes to move uh, a given amount of air. And it's resemble more those seen in obstructive lung disease because exhalation is more affected than inhalation. And yeah, this leads to a reduction in gas exchange surfaces, and this leads to an increase in respiratory rate. This is because this leads to increased dead space, since we have loss of gas exchange surfaces. O2 trapped, CO2 trapped due to lack of elastic recoil, and respiratory rate increases to maintain blood partial O2 and reduce blood partial CO2. the chemoeffective reflexes. Let's talk about gas transport and exchange, which is, I guess, more on the tissue level. Well, now the partial pressure is a pressure exerted by a certain gas within a mixture of gases. Dalton's law states that each molecule contributes to the same amount of pressure, no matter its composition. And the partial pressures are different in alveoli than obviously compared to outside. And the amount of dissolved gas in the solution is proportional to the partial pressure of the gas in the solution. So let's say we also have Henry's law. So Dalton's law was all are same. Henry's law is the concentration of gas proportional to the partial pressure of the gas. So let's say the concentration of the gas would equal to the partial pressure of the gas times the solubility, meaning how well they are dissolved. For example, under pressure, mm, under pressure, let's say the gas molecules are going to have less volume to deal with generally. If you imagine like squishing a balloon, there's going to be a lot of greater pressure, but there's also going to be a higher concentration. So because we have higher pressure, you have higher concentration, and you can imagine it kind of with lower volume as well. So yeah, these two laws are just something verbally you might want to understand. External respiration involves diffusion of gases between blood and air across the alveolar and capillary surfaces. So let's just draw it out. Ex External respiration is, let's say we have blood, we have these particles, and this is an alveolus. And O2 is going into the alveolus. CO2 exits blood to maintain this effect. Internal respiration is between blood and the interstitial. Let's say this is the body. Let's say this is muscle or something. Now we're going O2 to the muscle, and this would be internal respiration. And yeah, it's able to diffuse down to the oxygen gradient, and this is referred to as a gas transport cascade, where ventilation leads to O2 from the pulmonary diffusion, and this is circulating O2 for circulation, delivery, and to the O2 to the muscle, and then it's going to be O2 used up. And the rate of diffusion is affected by the pressure gradient, the surface area, and the thickness of the barrier, and this is known as Fick's law. So the rate of diffusion is equal to a constant K times, I should say the rate R times area for gas exchange. And this is the pressure differences over the distance, the thickness of the barrier. And in resting states, usually blood gas composition reaches equilibrium before it exits the capillary bed. This maintains. However, pressure gradients change during exercise or anatomy is the case. We won't see this. So respiratory reflexes can increase the rate of gas exchange. You could first change the pressure gradients here. By increasing the respiratory rate, you increase the exchange, and this refreshes the partial pressure gradients, leading to a greater rate of diffusion. And respiratory reflexes can also increase the rate of gas exchange by increasing the functional surface area. This is like breathing more deeper. You can increase the surface area and whatnot. And this allows for further inflation, increasing it for diffusion and whatnot. And this kind of goes into our idea of, I guess, our idea of anatomic dead space, where you might need deeper breaths to get rid of trapped air in this case. And gases must be transported through blood to reach tissues. And an important thing is remember hemoglobin in the blood units? Hemoglobin with their 4 heme unit, 4 globulin chain, 4 iron, that each binds to O2, and you can so binds to 4 O2 units. Well, all oxygen is carried by these hemoglobins. And yeah, and they're bound to these cells. And we have these cases of hemoglobin saturation curves. So we can have the pressure. partial pressure of oxygen, and we can also have saturation percentage. So it's saying at a given oxygen percentage, how much is hemoglobin willing to bind to oxygen? Hemoglobin, you exhibit cooperative binding. So if one of the four subunits have bound O2, the others become more likely to bind. This means that saturation is relatively high at rest. And when it's active, when it's metabolically active, so we have less PO2, hemoglobin offload more oxygen, which means the curve is going to shift downwards. Or wait, yeah, it's going to shift to the right. It's going to look like this. So this is going to be decreasing PO2 when they're anabolically active. This means we're going to have extra offloading of oxygen by hemoglobin, which leads to a substantial flow. It also has the same effect when blood becomes or tissues become acidic. This favors greater offload as well, and this is called a Bohr effect. And also when we have increased temperature. And you can imagine that the opposite happens here, where if you have increased PO2, increased pH, and decreased temperature, you're going to have greater hemoglobin binding, which doesn't favor oxygen upload. And like oxygen, most CO2 transport in the blood is managed by the red blood cells. Uh, CO2 can be bound to hemoglobin, and this can be converted to carbonic acid. So CO2 plus H2O turns into H2CO3. Then it's HCO3 negative plus H plus. This is converted via carbonic anhydrase, and it's reversible. And the H ions formed by carbonic acid can be dissociated to, can also be transported bound to hemoglobin. Uh, and yeah, this buffers the solution against changes in pH and favors offloading. So from tissue to blood, I guess the best diagram that you can really imagine is this. I draw it out for you. So if you have tissues that have CO2 and O2 entering, CO2 is turned out, CO2 plus H2O is going to be shifted from HCO3 plus H plus. And then the CO2 can move into blood, this is our blood cells, and the CO2 plus H2O H plus plus HCO3. And then the H ions in blood is bound to HbO2, hemoglobin plus O2, and this turns into HbH. So the hydrogen ion binds to hemoglobin, whereas O2, oxygen, is offloaded and is sent back to the tissue. And hemoglobin, there's also stuff with equilibriums. You mentioned that it pushes it to the right, favoring binding O2. So first, CO2 diffuses into the plasma or the red blood cells, into the plasma of red blood cells. And what's inside red blood cells? Carbonic anhydrase catalyze reaction, moving CO2 to carbonic acid and hydrogen. And accumulation of hydrogen pushes the equilibrium to the right, it favors H binding to hemoglobin binding to hydrogen and buffering our red blood cell pH. And O2 dissolves down to partial gradients into the tissues. CO2 is transported in primarily three ways in the bloodstream and removed at the alveoli. One is it's dissolved in solution, which is in this case plasma. Two is bound to hemoglobin. And three is it's converted to carbonic acid and bicarbonate plus H plus. And all these are easily reversible at the alveoli with a pressure gradient that favors CO2 movement into the air. Now, this is going to be it for respiratory. It's going to be a relatively shorter unit. We'll tackle immune and digestive and lymphatic system. Thank you for listening.